Okay, everybody. Thank you for the invite. I'm going to have to be a little bit selective in what I talk about, but hopefully, I'm going to talk a little bit about what fungi are, the past, the present, and possibly what the future may hold. So, there's a few things I've had to leave out, but unfortunately, given the time constraints, I've had to be, as I said, very selective. You can see from this picture, this is what most people think of a type of fungus. This is dryad saddle. It's a lovely one, squamous polyporosa, but hopefully I'll show you that these are nice mushrooms to look at, but the real dangerous ones are considerably smaller. Has that moved on, Steve? Yeah. So what are fungi? Well, they're eukaryotic organisms and were originally classified as plants. And this didn't really change till as late as 1969. And it's only recently analysis shows that 50% of the DNA is shared with humans. And this can cause a problem with the development of antifungals because of this sharement of DNA. It's easier to produce antibiotics against bacteria because their unique structures are not shared with us. But fungi, unfortunately, share numerous features. There are differences which put them in their own class. And one of them is a cell wall is made of chitin or chitin glucan complex. And it's not cellulose, which is found in plants. And chitin is the same compound, polysaccharide compound, that's found in insect exoskeleton. In our membranes, we have cholesterol, which is necessary to make them fluid and dynamic. But in fungi, it's replaced by ergosterol. Unfortunately, this can be used as one of the targets for antifungals. They lack chlorophyll, so they don't make any type of their own food. So you have to get the food by basically digesting anything outside of the fungi by complex enzymes, and they absorb it back into the mycelium. In 1969, when they decided things weren't quite right with the original classification system, what you have to remember, the Linnaean system originally only had two. It was animal and plants. But this chap, Robert Whittaker, in 1969, proposed a 5 classification system. So we've got the monera, which is basically bacteria, protists, fungi, animals, and plants. And it was him when he suggested that fungi need to go in their own kingdom. And some people used to refer to fungi as the 5th kingdom. In fact, there's a well-known academic textbook by a Canadian mycologist, and it's actually called the 5th kingdom. Now, that's all well and good, but with the advent of DNA sequencing and modern molecular biology, things have changed a little bit. And people keep trying to redefine the tree of life. And obviously, we've got what we used to be referred as the last universal common ancestor called Luca down here. And then everything arose from this ancestor. And you can see now there's a slightly different classification system. This is a simplistic one, by the way. It's got even more considerably complicated than this. But now the monera are broken down into bacteria and archaea. And then these are both branches of the prokaryotes. And then we've got the eukaryotes. And you can see plants, animals, and fungi branched off roughly down here. When this branching occurred, nobody really knows. And we don't really know how long fungi have been around for. The estimates range from over 1 billion down to about 500 million. But there's certain definitive evidence that they are around at the Cambrian explosion. And we think it's roughly round about there where the fungi diversified from the animals. And you can see they branch off here. Plants branched off at a different place. So this is why animals and fungi are closely related. Now, we're not quite sure how many fungi there are. Strictly speaking, no one knows. But in May 2022, there was a consensus that suggests there could be as many as 6.2 million species. Some people argue that that's too much. Some people even say it could be considerably more. But in comparison, that's 16 times more than plants. And also, the current estimate is only of 7.77 million species of animals, and most of them are insects. So fungi are a very prodigious group of organisms. But out of this possible 6.2 million species, only 155,000 are currently named. We know for a fact that 300 of these fungi cause diseases, but they're in capitals at the moment because there is a problem at the moment with climate change, which I'll move on to as the talk goes on. But this looks like it could be a serious problem in the future. But as regards 300 in human disease, more alarmingly, fungal diseases are responsible for most plant diseases, and they think it could be as high 85% of all plant diseases have a fungal cause. And at the moment, there are over 8,000 species of fungi and fungal-like organisms that we know can cause plant disease. Now, I don't want to dwell on this, but this is the current state of affairs as regards fungal phylogeny. Most people are familiar with the Ascomycetes and the Basidiomycetes, but it's now got more complicated. These are the rusts and smuts, and they belong to Basidios. But we've also got these new divisions of Muromycota, Zoopagamita, Chytridio, and Blastoclidio, and Orthosporidia. This is, we think, the current state of art. Whereas again, with DNA sequencing, this all could change by next year or even by next week. So fungal phylogeny is very, very complicated at the moment. But because of that diversity of classification, it leads to a diversity of fungal morphology. We're all familiar with our familiar fly agaric, the typical mushroom that's featured on most covers of kids' books and magazines, and what we see on public imagination. We got the oysters, brain fungus, coral, but turkey tail. But most fungi are microscopic, and these are scanning electron micrographs of a huge magnification showing different types of fungi. And another one you're probably familiar with is if you leave fruit or bread around for a while in damp, humid conditions, you get a growth of fungal on top of fruit. And this is a major concern especially in developing world, of fruit rot. So there's a huge diversity of fungi. Now, where are they found? Well, it's been recently analysed that if you just do a random location, a sample in the air, even at 30,000 feet, roughly where air travel occurs, you'll pick up fungal spores. And I think this is one of the main routes of distribution or contamination from one continent to another. One of their most ubiquitous sites in soil, and that's where a lot of our samples are currently being used to try and develop new cancer treatments and new antifungals. We know and we've heard of mycorrhiza, especially in the press, and these are the fungal associations with tree roots and plant roots. And it's been said that without these, plants and flowers would not really develop to their full capability. And it's because of these mycorrhiza that plants can get their proper amount of minerals and water allowance from the surrounding environment. This is recently been found. Endophytes are found inside plants and trees, especially within the very thin layers of leaves themselves. And there's some indication that these may be involved, especially in the production of what we call secondary metabolites that are involved in communication between plants and trees, and also deterrents for insect attack and herbivores. We now know they're actually found in water, both in freshwater and marine. We found them on rocks. They're an important component of the microbiome not just in our situation, but also in animals. And there's a class of fungi from the chytrids, a subphylum of the chytrids, that are responsible for the breakdown of cellulose in herbivore ruminants in the stomach. We know they're symbionts, especially lichens, and because of this, they can therefore colonize Antarctic and Arctic. They're responsible for producing original colonizers on land and actually starting the process of making soil. And this is a new one, disaster sites. We're now aware that after a major disaster, some of the first organisms that come in, and a problem is with them causing disease, is at disaster sites. And I'll touch on this in a bit later. But somebody has suggested recently that we look at a new branch of microbiology called disaster site microbiology. And there's quite a few people have actually said this needs to be taken more seriously. And in fact, there was Hurricane Katrina, I think it was 2009. It obviously you've seen on the news the disaster it caused, but apparently there was about 194,000 homes that were affected with mold coming in after disaster, because obviously these tornadoes occur in sort of wet, tropical, semi-tropical, hot, humid environments, and these are perfect growth conditions for fungi. So 194,000 homes were affected, and they reckon about 70,000 were actually condemned. And as of October 2022, it's estimated that $142.9 million has been allocated to alleviating some of this burden. But this has only covered about 92,000 houses so far. So this is a huge economic burden. So what do they do? Well, they're called the great decomposers and recyclers. And people have said that if we didn't have fungi, then we would be up to our necks in rubbish. They actually decompose, especially plant material, animal carcasses, and they recycle those nutrients back into the environment. They've now been shown that they're incredible at carbon locking mechanism. They produce medicines, which I'll go into a bit later. Food, they're actually involved in the production of food, such as corn and cheeses, but they're also a good economic prospect in themselves. So fungi, as we see on the supermarket shelves, is a multi-billion-dollar industry. And obviously the one that most people try and find is the truffle. And it's very, very competitive in places like Umbria in Italy. And in fact, I can't remember the year, but Sotheby's auctioned a white truffle, which weighed just over 4 pounds, and it sold for $60,000. So it's a very the trouble is people try to grow truffles artificially, but it doesn't seem to work. They've been used for numerous years as a model organism for study of what we call cell cycle control, especially mitosis and meiosis and cancer. And we mainly use yeast for this, especially Saccharomyces. But the beauty of this is because they grow in culture and grow very quickly and produce numerous generations, you can knock out genes and see what the effect is on cell cycle control and development of cancer. We use them in biotechnology and agriculture. Because we can grow them in culture, we can modify the DNA so they can produce products that they don't normally produce, but they can produce them in huge amounts. We also are now looking at biomaterials as the future. And I'll give you an example at the end. And a hot topic at the moment is bioremediation. And this is using biological control to try and decontaminate sites. But fungi are mostly involved in mycoremediation, and I'll just touch on that at the end. So we'll start with something slightly controversial. This lady down here is Susan Simard. She's now Professor of Forest Ecology in Canada. And in 1997, she produced a rather dry sounding paper called Net Transfer of Carbon between Tree Species with Shared Ectomycorrhizal Fungi. And this was published in Nature. Now, everybody who's a scientist, their aim is to get something published in Nature. And if you can get yourself featured on the front cover of Nature, you know you've made it. And this is Susan down here. She produced a fascinating book called Finding the Mother Tree. Now, there's a lot of controversy at the moment. We know ectomycorrhizal fungi exist. We know what they do. But what they're saying is she tended to over-exaggerate her findings. Now, to get published in Nature, it's all peer-reviewed. It has to be rigorous science, numerous controls. And this wasn't just a paper out of the blue. This was based on years and years of research, controlled studies, and numerous. She did the experiments numerous times just to make sure that it just wasn't a fluke. She's now being criticised by fellow scientists. The public like the idea of plants talking to each other, but I think that the problem is it's got a little bit out of hand, because once the public get involved and social media, then what they've tried to do is say plants and trees are making a conscious decision to do these things, where it's basically just nature working as it should do. But what nature did, a lot of people attribute this name, the Wood Wide Web, to Susan, but she didn't actually coin it. It was the editors at Nature who found out that this was a nice way of saying what these findings were. One of her team that used to work with her has criticised her as well, but it's now been found out she's been funded by the Canadian the Tar Sands Foundation. And one of the criticisms actually said that her findings were statistically significant, but not biologically significant. Now, if you've ever heard an oxymoronic statement before and you want an example of one, then there you are. I like the idea, and numerous people like it as well, especially Melvin Sheldrake, if you've read his book. So medicines, you're probably all aware of penicillin. Now, Alexander Fleming in 1928 was using bacterial cultures, and he left the window open, went on holiday for a weekend, came back and noticed there was growth on his plate, but only in certain areas. There's some debate now about whether it came in through the window or if it came from the mycology department underneath his floor and it escaped through their window and up into his. But it doesn't matter, whatever happened, we know it was serendipity, so it's an accident, and it produced penicillium from the fungus Penicillium. He was awarded the Nobel Prize along with Ernest Chain and Howard Florey because they actually isolated the compound. And because of penicillin, it's estimated that only 1 in 15 soldiers died in the Second World War of disease, and it saved numerous lives because most of them actually died in action. But before the advent of penicillin, any injury or infection was not guaranteed to be fatal, but it wasn't far off. So this is penicillium growing in culture on an agar plate. This is what it looks like under the microscope. These are conidiphores, and these are where the spores produced, and this is microscopic, and this is what produces penicillin. And if you grow bacteria on a plate, you can see it grows. This is where it's been streaked on a plate. But if you have a culture of penicillium on this plate, you can see you get what's called a zone of inhibition. So we know that the fungi is producing something that is inhibiting growth of a bacterial colony. And this, in this instance, it was proved to be penicillin. Now, I don't know how many of you are on statins. If you're not on them yet, you might be prescribed them soon. It's probably the most prescribed drug in the world at the moment. And it was discovered in the 1670s by a Japanese scientist called Akira Endo. And he discovered that it was a relative of the penicillium that made penicillin, this one, Penicillium citrinum, that it made a compound with cholesterol lowering properties. Other scientists thought, well, if it's one penicillium, then it could be found in others. And they found the same properties in Aspergillus terrace and also in other fungal species. And this has led to the development of a wide variety of statins. Atorvastatin is one of the ones that's commonly prescribed. But in the early days, it was Lipitor, which is lovastatin. And this was the first one made. Now, in its heyday, it made $12 billion a year. So this is why people are looking at possible new molecules from fungi. Now, this scientist was on holiday in Norway. He worked in Switzerland, and this was in 1969. And while he was on holiday, like good naturalist, he collected 50 samples from different areas, mainly soil, and he took them back to Switzerland for analysis. And most of them proved useless, but there was one isolate called Tolypocladium inflatum, and what he noticed, it suppressed immune system from mammals with very, very little toxicity. So this was a really, really nice finding. And this led to the development of cyclosporin A. And we now know that this works by the mechanism of inhibiting the synthesis of a group of compounds called interleukins, especially interleukin 2. And these interleukins are essential for the self-activation of lymphocytes and also their differentiation, such as into killer cells, memory cells, and cytotoxic cells. And as we say, the rest is history. And what I mean by that is, before the development of this, transplantation was quite a risky and more of an experimental procedure than of a life-saving procedure it is today. And cyclosporin A changed this literally overnight, and it made it from an experiment procedure into a routine operation. And it's just thanks to this sort of almost accidental discovery, this compound from this fungus. For those geeks out there, if you want to know what cyclosporin looks like, it's a very, very complicated ring structure. Its formula is C62, H111, N11012. It's got a large molecular weight, but unusually for a compound, it's actually made of 11 amino acids, which is quite unusual. But to synthesize this in the lab is, I think, don't quote me on this, but I think it's impossible. So they actually grow it in culture, and this is the only way you can produce enough cyclosporin A to make it viable for transplants. Now, the controversial topic is psychoactive mushrooms. Now, in recent years, the use of psilocybin mushrooms, especially psilocybe cubensis, which is highlighted here. Most people know this as the Liberty Cap or magic mushroom. You might have seen people on lawns or out in grassland trying to find these. And it produces the psychoactive compound psilocybin, which is a relative of LSD. And obviously, because of social media, the press, and probably you might know people, I'm not saying you do, but it's well known for its recreational use, especially due to its psychoactive effects, especially hallucinations. It's also been shown to produce euphoria, and some people have used it for mystical enlightenment. Now, research was done on this many, many years ago, but in the late 60s, it was banned. But recently, people have started to go, oh, hang on, let's go back and look at the original research and perhaps do some more control studies. And these have been done, and now it's been now scientifically shown and proven, apart from the anecdotal usage where people have said it's helped me conquer all these disorders. We now know it's been shown conclusively that psilocybin can treat conditions such as depression, anxiety, addiction, and post-traumatic disorder. We also know it's been shown to help treat terminal care patients. Aldous Huxley, some of you might know, wrote a couple of famous books, Brave New World, and I forgot the other one. Oh, Roger might know it. But on his deathbed, he asked his wife to inject him with LSD and psilocybin. It was a great anecdote of mescaline, by the way, but he showed that when he was dying, the use of these psychoactive compounds made him at peace, and he died a very, very peaceful death. Apparently, he was tripping at the same time, but he didn't care. In New York University, a small study has shown that just 2 doses with therapy, with psychotherapy, has actually helped people break alcohol addiction. And a group at Johns Hopkins in America are now analysing the use of psilocybin for anorexia, obsessive compulsive disorder, chronic Lyme disease, and cognitive impairment. And in 2023, 2 states in North America, Oregon and Colorado, actually legalised the use of psilocybin by adults. Now, some of you might remember this name, David Nutt. He was sometimes referred to as the drug czar, and he's absolutely brilliant. He talks a lot of common sense, but because his use of common sense, he was sacked by the government at the time. I think this was the late 90s. He's now Professor of Neuropsychopharmacology at Imperial College London, and he's recently published a paper and stating that when coupled with psychological support, effects can produce a rapid improvement in mood among people with depression that can last for months. He's published numerous papers, and I think he got in trouble because he basically said that ecstasy and LSD was nowhere near dangerous as alcohol and tobacco, and he's actually right. But the government don't like that, and most British governments are, as I'm concerned, a bunch of cowards when it comes to looking at the uses of these psychoactive drugs. The trouble is, people will use them anyway, and people say it makes a massive difference to their lives. One of the big groups of fungi I want to have a look at is called Aspergillus. We now know there's about 339 species. They're particularly found in soil, but its use in biotechnology is related to production of soy sauce, enzymes. It's involved in a huge production of citric acid, and as I said, it produces statins. Now, they're not considered to be a major cause of plant disease, but they are responsible for several disorders in various plants and plant products, especially when these plant products are stored. And the notable consequence is when storage of food is kept under the wrong conditions. And when Aspergillus grows on this food, it can produce small compounds which are extremely dangerous, and they're called mycotoxins. And the mycotoxins that are found in Aspergillus are aflatoxins, ochratoxin, and to a lesser extent, fumonisins. Aflatoxin B1 is the one that's the most carcinogenic. Now, we know it can cause a spectrum of infections, mainly due to 4 species, Fumigatus, Pteris flavus, and Niger. Now, most fungi can be fought off by most people. Fungi tend to be a problem in immunocompromised people, and it can be people who are on drugs for transplant. It can be cancer, it can be diabetes, or it can be conditions like AIDS. But most people can fight off these syndromes. But clinicals, the nature of the disease will depend. Now, if it's left untreated, these conditions can reach 100% mortality. You can get more indolent forms. They can form chronic pulmonary aspergillosis, where these fungi actually grow within lung tissue. but they can produce allergic reactions as well. It's often with people with underlying lung disease themselves. Now, in the early 1960s, the discovery of mycotoxins, especially aflatoxin, began when there was an outbreak of death in 100,000 turkey poults. They couldn't find any reason at all for why these young turkeys died, so it was called Turkey X disease. Fortunately, there was a veterinarian at the time called William Percy Blount, and this is him on the left, and he started investigating them, and he noticed that most of them suffered from liver damage, especially with cancer. And under investigation, he found that the disease was associated with the meals that were being fed to these turkeys. And it was mainly Brazilian groundnuts, and they were affected by Aspergillus flavus. And the toxin from this was called aflatoxin. And this is where basically this man was influential in looking at production of mycotoxins and how they affected not only animals but humans. And we now know that contamination by aflatoxins is a global health problem. And even with our knowledge now, it's still a serious problem, especially in developing countries. And again, it's mainly in tropical countries with good humid conditions. Now, I managed to get hold of this. This hasn't actually been published yet. It's a preprint. But scientists at Manchester University have used computer models and simulations to look at how Aspergillus might be changing over the years. And they have suggested that their models aligned with climate change will allow certain Aspergillus species, not all of them, to spread across North America, Europe, China, and Russia. And one particular one, Aspergillus fumigatus, is expected to move northward towards the Arctic, with its range increasing by up to nearly 80% by 2100. And they reckon this could potentially lead to an additional 9 million Europeans being exposed to this fungus with corresponding disease. Now, this might sound bizarre, and I'll try and explain this, but in contrast with some regions like parts of sub-Saharan Africa, they may actually be getting too hot for Aspergillus to actually function and grow. Now, some people may say, well, that's a good thing, but Aspergillus is an important part of the environment. And as if you know, if you disturb one aspect of an ecological environment, it has numerous consequences up the ladder from where they grow. And most people don't realize that the fungi grows roughly at an optimum of 25 to 30 degrees. So they're thinking at the moment with climate change and the rapid growth of fungi, they may become what's called thermotolerant. So they may actually allow this growth into northern temperate regions. But if it gets too hot, it may actually inhibit the growth of Aspergillus and other fungi. So you could even lose the mycorrhizal association with trees and plants. We don't know. It's a great unknown, but it's something certainly to think about and worry about. This is Aspergillus. Again, it looks like penicillium. Most of these microscopic fungi have a very similar structure, and they're very difficult to identify microscopically. Usually you have to grow them on a selected agar medium or do DNA sequencing. This is it growing on maize, but this is it grown on peanuts. And this is what it looks like on stored peanuts. And it's just this simple growth that produces those mycotoxins, which lead to this liver cancer. Now, Aspergillus grows on the crops. They can then produce animal feed. It gets into animal feed. And what has just been realized is one of these aflatoxins can be converted into another one that's then found in milk. It can get direct contact with animals and humans. It can go into food production. But whatever happens, contamination and contact and exposure can lead to what we call acute aflatoxicosis. It can lead to immune suppression. It can lead to growth failure in children. But importantly, it quite often leads to the development of liver cancer. Now, I'll move on to another condition called white nose syndrome. It was noticed in 2006 that thousands and thousands were hibernating bats were suddenly dying. And this was first seen in a cave in Albany near New York. And when they looked closely, they noticed the bats had a white substance, mainly on the noses, but when they looked closely, they're actually on the wings. And they didn't know at the time, but this was a suggestion that there could be a fungus involved. And it took 3 years later before they identified the fungus, and it was a brand new species, Pseudogymnoascus destructans. And I think this part of the name gives it a bit of away. So that was 2006. 2009, they identified it. By 2009, it had spread to Virginia. By 2022, it was then in 38 states and 9 Canadian provinces. So it had spread. This, I don't think it's that clear, but each one of these is a dead bat. So can you imagine tens of thousands of these just being found on a cave floor? It's not so obvious here, but I can't remember what the species this is. It doesn't really matter. It's an American species, but you can see the white nose where it gets its name from. So this is fungi growing on the nose of these bats, and you can just see the white bits on the wings. So it actually interferes with their flight mechanism. We know at the moment there's 12 North American bat species impacted, including one that's been endangered for years and is now on fear of going extinction, and that's the northern long-eared bat. They've found that in some caves there's been a 90% reduction in bats within these caves. And the US Geological Survey estimates that bats contribute to more than $3 billion to the economy in pest control, seed dispersal and pollination. So if you're going to lose this amount of bats, the economical impacts are going to be huge. Nobody knows how it spread. We think it was somebody from Europe who may have brought it over on the clothes, the shoes, and introduced it into one cave, and then it spread. We don't know the true number of the bat deaths, but in 2012, it was estimated there were 6.7 million bat deaths. Now, what they're going to do about this? What they've tried to do is vaccination of bats. Well, it's very, very difficult to produce a antifungal vaccination program. It's almost impossible. So what they've tried to do is develop a gel that you can spread on bats, because bats are very good at social grooming and they will lick each other. That was one idea, was to coat some bats. The other idea is, as I was telling you about fungi, they only grow at a certain temperature. So the idea was to warm bats up. And they did a controlled experiment where they actually took a subpopulation, a small population of bats, and warmed them up. And it didn't result in 100%, but most of them didn't get white nose syndrome because the fungi couldn't grow at the temperature the bats were. The trouble is, if you start warming bats up in winter, they're then going to come out early where there's no food for them. So it's a problem at the moment. What do you do in this situation? Now, we know that Britain has suffered, well, Britain and America suffered from elm dieback mainly in the 60s. And elm dieback is still a problem, but it doesn't seem to be talked about at the moment. The one in that's talked about at the moment is ash dieback. and it was formerly called Cholera dieback. But we now realise there's actually two stages to the fungal growth. And the fungus was first described in 2006 under the name Cholera fraxinea, which is how it got the name Cholera dieback. But it took another 4 years before it was discovered that Cholera fraxinea was actually the asexual stage of the fungus. And it's this stage that does the damage. But the fungus is now called Hymenoscyphus fraxineus, and it's an ascomycete fungus. And this is the fungus when it's in its fruiting stage. And it's this stage that has given name to the fungal cause of the disease itself. Now, ash dieback, this is a relatively healthy ash. This is what it looks like when it's got dieback. This is the fruiting body of the sexual stage. You can see this is just a twig, so you can see how small it is. I'll explain the significance of that in a minute. But the disease is now widespread in Europe, and they think at the moment there's an 85% mortality rate. Some people think it could go as high as 95, and some people are even talking about 99. But in plantations at the moment, it's high because they're planted next to each other, but they think it's, well, only 70% in woodlands. The fungus that causes the disease is actually related to a native fungus, albidus, which is harmless to European ash trees itself, but it's this other fungus that seems to be causing the problem. And there's growing concern from 2016 in Journal of Ecology, which warned that in combination with Fraxineus mushroom and the emerald ash borer attack, which is now in this country, it could actually result in the actual extinction of ash trees. If you look at ash dieback, it first appeared in Poland, but we didn't first find it in this country till 2012, but there is some anecdotal evidence that it could have been here since 2006. We don't know, but it was first noticed in 2012. But you can see it's now spread nearly all across Europe. It doesn't seem to have been in America yet. It's just mainly European ash that seems to be the problem. And if you look at the dieback in the UK, you can see, I know it's not a very good diagram, but you can see that the majority of the UK is affected by ash dieback. There's remote areas of Scotland which still seem to have escaped it, whether that's because there's very little ash there, which is probably the reason, but basically the entire area of UK is affected by ash dieback. Now, the spores are airborne. They seem to land on keys, ash keys, or leaves themselves. These then grow, and it's this growth of these spores in the asexual phase that produce the dieback. These leaves and keys then fall onto the ground. The spores then go into the sexual stage, and in the sexual stage, new spores are produced, and they can be released back into the environment, where they can go back and infect the same tree. They can go infect another tree, or it's now been shown they can directly infect roots of the same tree or neighbouring trees and saplings. So it's a process which is probably impossible to control. They're actually relying on the fact that some trees are starting to show resistance, but saplings seem to have succumbed to disease within a couple of years. Big mature trees, they think it can take anywhere between 10, 20 and 30 years to show evidence. So we're not quite sure yet what the future is. But there is a program of trying to breed at the moment resistant ash. Now, this has just been published. Oxford University estimates that just from this fungus alone, up to 99% of all ash trees could die. It's also looked at the economic aspects, and this includes removal of dead waste, carbon locking, uh, flood protection, and they think it could cost up to £15 billion to the UK economy. They also warn at the moment there's another potential 47 other known tree pests that could add another billion a year to costs. And this is interesting. In 2007, the annual value in trade in live plants was £300 million in the UK. But the government decided that this was a valuable trade mechanism and biosecurity was, I wouldn't say lapsed, but it wasn't as secure as it should have been. And obviously biosecurity lost out to the economics of trade. And at the moment, this is probably an area which is costing the UK a serious economic loss in plants, because I think there's going to be more coming into this country in the future. Now, this is a hell of a statement, and this was written in the Science magazine in 2019. Science magazine is sort of the rival to Nature, so it's very high up there in its opinion. And the statement was the greatest recorded loss of biodiversity attributable to a disease. And basically what they were saying, this is the massive die-off of frog species. We now know that Batrachochytrium dendrobatidis is the fungal species responsible for disease, and the disease is now called chytridiomycosis. At the moment, it's found on all continents except Antarctica. So it's making it a huge global threat to amphibian biodiversity. And currently, we know it's driven the decline of at least, this is conservative, this is at least 501 amphibian species worldwide. And 90 species of frog are now considered extinct or presumed extinct. Because they've tried to find these species and they haven't found them, they're actually now having to assume, because they haven't found them, they're presumed extinct. I mean, there could be isolated populations in remote areas of tropical rainforest, but they're now assuming 90% species of frogs could be considered extinct. Another 124 species have declined by 90% or more, and all these declines except one are caused by this single species of fungi, which is, rather than saying this all the while, we'll just refer to it as BD. Now, where it came from, nobody knows. One paper has historically linked it to infection in amphibians in South Africa since 1938. Other people think it may have come from Korea, a peninsula on Korea. We're still not absolutely sure, but late 20th century records actually started to show declines in Australia, Central and South America. The climate change was originally blamed, because you can blame climate change for everything. But in 1997, the fungus was identified, and in 1999, BD was named as the cause. Like bats, they've actually come up with a solution to try and warm frogs. And in a controlled experiment, if you warm frogs up, in one experiment, all but one died. So it does work. But if these fungi become more, they start to grow, become thermotolerant. Now you might think, well, if they're in equatorial regions, they are thermotolerant anyway. Most of these are in high altitude, so they are in wet, humid conditions. Now, collateral damage, if you haven't got frogs controlling an insect population, especially Anopheles, mosquitoes, you're going to get malaria. And we know for a fact malaria has started to increase. So again, if you disturb one part of the ecosystem, it has massive knock-on effects on other areas. How does it work? The fungus is tiny. We're probably looking at anywhere between 2 and 5 microns. So that's 2 to 5 thousandths of a millimeter. And these zoospores They are flagellated. Chytrids, by the way, I think they may be the ancient fungi, and they think all fungi may possibly have come from a chytrid, because most fungal spores do not have this flagellum. They've lost it. But in chytrids, they have this flagellum, which makes them very motile in water environments. So they latch onto the skin of a frog. You get the invasion from the germ tube. They then start to multiply. You then get an immunological reaction. They actually multiply within the skin. You get skin thickening due to the fungi. You get skin thickening because of the cells of the epidermis start to thicken themselves. That's called hypertrophy. And then the cell numbers actually increase. So you get an increase in keratin. So you get hypertrophy as well as an increase in numbers, hyperproliferation. And not only with that, but you get damage to the cell surface, which then leads bacteria to be able to invade the cell surface. And also you get skin sloughing off as well. And I don't know if most people realize, but frogs actually breathe through the skin. It's very, very thin. So basically, with an increase in cell proliferation and cell numbers and cell size, they aren't allow themselves to breathe, and effectively they die by suffocation. But you can see it's a very quick process from initial invasion up to full thickness, hyperkeratinization, and sloughing the skin. It's only 4 days. This is what a frog looks like when the skin starts to slough off, and this allows bacteria to come in, and this is the death sentence. Now, that was frogs, but what they've noticed recently is a fungus similar to BD, another Batrachochytridium, has been killing salamanders. It's found mainly in Europe at the moment, and it's currently been blamed for wiping out 96% of fire salamanders in the Netherlands. It's not in North America yet, but North America is starting to panic. So what they've done a laboratory study to see if this fungus will actually infect and do damage to North American species. So they looked at the effects of BSL on 35 salamander species from North America, and they found it caused infection in 74% of species with a mortality rate of 35%. So this is a warning. Now, whether it will cross over to North America, we don't know. But looking at other diseases, I can't see why it won't happen. Now, this has also set alarm bells ringing, because studies have also found that this chytrid fungus, the one that affects frogs, can also infect some group of organisms called caecilians, actually in wild activity, and cause mortality. If you don't know what these are, they look like snakes, but they're limbless amphibians. They're only a small group, but again, because of this fungus, it could cause a huge decrease and even extinction in this group of organisms. Some of you, if you were born early on in life, from the 1920s, there was a song called "Yes, We Have No Bananas." I've used this as a title because bananas are seriously in trouble at the moment. Now, in the early 1900s, there was only a single variety of banana that dominated the banana industry, and that was called the Gros Michael. Now, in 1920s, a fungus called Fusarium oxysporum was first seen in Suriname, and then it started to infect harvests in other Caribbean countries. And in 1960s, it wiped out almost all Gros Michael bananas. And the problem is, all these bananas were a single clone. So there was no genetic diversity at all within these bananas. You can still buy these Gros Michael bananas, because some of them have been grown in very, very small populations throughout the world, and they weren't affected. But it was a problem. So what they had to do was then find a solution. So they produced another banana called the Cavendish. And this was produced because it's resistant to Fusarium. And this has become the dominant supermarket product. But guess what? As you may know, the Cavendish is in trouble. A new relative of the original Fusarium fungus called tropical race 4 was first identified in Taiwan in 1990s. It's now spread to Asia, other parts of Asia and Africa. But in 2019, it was found in Colombia, and Colombia is considered the banana capital of the world. And the UN has now warned that banana production is now threatened. So we could have another gross Michael on our hands. And again, Cavendish was a clone. So we didn't learn from the first problem with the gross Michael. So we produced another clone. So rather than having multiple bananas from genetic diversity, we had another clone up. So the problem is now to find another banana. Now, human infections, when you tend to think of them, you tend to think of athlete's foot thrush, either oral or vaginal, or you think of common bathroom mold. But unfortunately, fungal pathogens themselves affect 1 billion people worldwide. That's a huge number. Again, it's mainly in developing countries or in immunocompromised people. And one particular problem is fungal keratitis. And this is an eye infection. And it's estimated that currently 1 million patients will actually go blind because of fungal keratitis. We know fungi produce allergic problems in susceptible people. It's estimated that currently it affects 10 million people. It's common in people with lung problems anyway, but with global warming, this could again become another problem, especially with Aspergillus, with that Manchester programme showing they're moving north. And serious fungal infections kill approximately 1.6 million people per year. And that's more than malaria itself, and it's equal to the number killed by tuberculosis. Now, this is just an example of what fungi can do. These are the fungi growing in culture. They're microscopic, but once they get inside human tissue, usually via a wound. Now, this is an example of a patient from Sudan. And in Sudan, it's a huge problem because of poverty. A lot of people haven't got access to protective gloves or shoes. And if you step on a fawn or catch a fawn in your hand, most of these fawns are covered in bacteria and fungi. And this is a fungus growing and destroying a hand and a foot in a patient in Sudan. Now, I could have shown a lot worse pictures than it, but Steve doesn't like seeing them. So I've kept it mild for Steve. But you can get these mycotomas where you get gross disfigurement of limbs and faces. And now these can be treated even at this stage, but because this is in a developing country, the access to antifungals is either minimal or absent. Now, ergot. Ergot is a fungal disease normally found on rye, but it can grow on related cereals. And it produces these growths called sclerotae or sclerotium. And it's these that have, is produced by a variant of claviceps fungus called claviceps purpurea. And this is the one that produces ergot. These sclerota then dry and fall off, or they can be harvested with the seeds. Now, when the seeds are collected and dried, it sometimes can be very difficult, especially when these are small, to differentiate between the two. And this is how it gets into the food train. And once these seeds are ground up and produce flour, you've still got these products released from the sclerota. And one of the compounds that produced is ergotamine. Now, ergotamine was famous in the old days for producing a condition called St. Anthony's fire. And it's actually produced by ingesting either contaminated seeds or the flour from grains that are infected with the claviceps fungi. The name gets its name from the Benedictine monks who worshipped St. Anthony, and they sometimes treated patients by using relics of the saint as a medicine. It gets its name fire from the reference to you get a burning sensation, especially in the extremities of the body. Now, we've got no figures at all to indicate what the numbers of people are infected, but there is one number, and this was an outbreak in France in 994, and we think it caused the deaths of 20,000 to 40,000 people in that country. Now, why it causes deaths is not because just you get the burning sensation, but it can induce gangrene, convulsions. You can get sores that then open and then allow bacteria or other fungus to come in. It can drive people mad, and it's plagued medieval society for a number of years. I mean, there was a miserable existence then, but this further exacerbated it. This is a calf that has ingested ergot contaminated food, and this is a man who has got ergot or ergotamine induced gangrene in the feet. That's a normalish hoof, but you can see gangrene just starting to spread into this one. So you can see the effects. Before I go on, ergotamine, I'm not sure if it's still used. I think it can be prescribed in very controlled doses for the treatment of migraine. There's newer drugs on the moment, but that's what its initial uses were. It can also be used in women who have had experienced very, very heavy bleeding during birth, because what it does, it closes down blood vessels, and that's one reason why you get gangrene. There is controversial evidence at the moment that it may be involved in the Salem witch trials, because there was a number of women, I think it was between 200 and 300, that were convicted and hung and burnt in Salem in Massachusetts in the late 1600s. And they suffered periods of convulsions, hallucinations, and wild dancing. And there's now evidence that it possibly could have been due to ergotamine poisoning. Now, I want to talk a little bit about this one. This is a problem. Most people have probably heard of Candida albicans, the yeast, and that's what causes oral thrush and vaginosis. Or in certain men, you can get what's called jock rot or jock itch. And those are fairly benign conditions, easily treatable. They can be invasive, and they can cause death in untreated people. But in sort of Western society, Candida albicans is okay. It's a problem, but it's not a major problem. But this one set alarm bells. In 2009, in a Japanese hospital, they isolated a new species of fungus from the external ear in a 70-year-old patient. And because it was a new fungus, but it was related to Candida albicans, it was called Candida auris. Auris from the ear, because that's where it was isolated from. Now, everybody said, okay, it's interesting, it's a nice case study, but it's probably an isolated case, nothing to worry about. But between 2012 and 2015, it was reported simultaneously in patients in South Africa, Venezuela, and India, who were all seriously ill. Not just ill, but seriously ill. And what they found was all the fungi, even though they all belong to this, were genetically distinct. So it wasn't a cause of the fungus moving. Something looks like it's happened that's caused this fungi to suddenly become virulent. The problem is it's very, very resistant to treatment, and 1 in 3 patients have died or will die. And as of 2015, most cases are resistant to all known fungal treatments that we have available. Now, the numbers are difficult to ascertain, but we think this fungus is now killing people in 40 countries on 6 continents. When people have Candida albicans, it's very easy to clear up. In hospitals, you can kill it with bleach. You get rid of it. You change the bed clothes. You wipe the beds down. It's fine. This one, you clean the beds down. You wipe everything down. You bleach it. It stays there. And it's been found to lie around on hard surfaces for months. Even when other fungal species have been cleared, it still lies there. And the World Health Organization in 2024 now consider it one of the most harmful fungi to humans. Yeah, it's just a variant on a yeast. And the question is, why? What suddenly caused it to change? And we think it's all due down to this fact that it's become thermotolerant. It's starting to grow at higher temperatures, and it's starting to grow at body temperature. And this is the problem. Most fungal species will not grow at a human temperature. Obviously, if you're immunocompromised, you've got a problem anyway. But this one, they think it's jumped and it's become thermotolerant. This is currently the list of human fungal infections. And you can see they all look similar, but they're all microscopic. Okay. Pneumocystis was the one that was found in AIDS patients, and that was one of the cause concerns, and that was how AIDS was identified, the AIDS complex in the early days. Cryptococcus, Histoplasma, Fusarium, Candida, Aspergillus, they're all now becoming a huge global risk. And for the World Health Organization to list 18 to 19 fungal species as a concern, then you know something's going wrong. Now, we've talked a lot about the bad side. What about the good side? Well, people are now trying to harness fungal as a material, and we've now got myco-leather. This is a piece of fabric produced by Micron. It's mainly produced by Ganoderma. You've probably seen that bracket grown on trees, but this is produced from Ganoderma. The beauty of this is you can dye it in various moods or colours. Stella McCartney's actually got involved with this as well. And I can't remember what she's produced, whether it's clothes or handbags, but there's a number of companies now selling products made from myco-leather. So this could be a revolutionary winner and hopefully turn people away from normal leather. We're also looking at using fungi for buildings, and this is a huge development that's going on, and it's called the Phoenix. It's going to be 300 units of affordable housing in Oakland, California. Oakland's quite a deprived part of California, and they're hoping it's going to help push fungi into the building industry. Now, it's not a house or a complex built of fungi. The core buildings will be made from a mycelium composite. At the moment, you can make mycelium bricks, but to build an actual solid big structure from mycelium, pure mycelium, is out of reach at the moment. But the research is going that way, so it could be the future. Now, this is not a very good slide, but it's the best one I could find. Hot topic at the moment is myco-remediation. We know we're polluting the world, and we're polluting it with biologically, chemical, industrial, and that can include nuclear waste. So what they're looking at is using fungi to help clear up the environment. And we know many fungi can feed on bacteria that are pathogenic to humans, so they can use that to clean up that. Can use it as a filter for filtering out wastewater. It can break down contaminants. And what we found out, there's a group of compounds called PAHs, polycyclic aromatic hydrocarbons, which are really nasty. But wood degrading fungi seem to be particularly good at breaking down PAHs. So hopefully that may be one area of look. And they're looking at using it on oil spills as well. It's even been shown to break down TNT and dioxanes as well. Living filters can capture hazardous site runoff. So again, possibly from paper mills, dye manufacture, and power plants. And what they found is at the moment, they've been looking at Chernobyl, and they've actually found there's over 200 species of fungi grown in Chernobyl, even inside the core itself. And what's unusual about these fungi is they all produce huge amounts of melanin. And there's possible evidence that you could use these fungi in a culture medium, produce industrial quantities of melanin. And you could even use these as protection suits. But they're wondering why these fungi are grown in Chernobyl. And they think the melanin may be acting very similar to chlorophyll as an energy trap. So rather than trapping sunlight, it's trapping gamma radiation and possibly using it as an energy source. It's making it into the press and films and video games. And this has been on the telly recently. It's a very successful television series. I haven't watched it. It's a video game that's very successful. It's called The Last of Us. And it's based on fungal infection of humans, turning them into zombies. Now, you may think that, well, that's never going to happen. Well, in nature, it already happens. And there's a species of fungi called Orthocordyceps, and it infects ants. It can infect other things, but the main one is ants. And it produces these huge bodies up here. Now, to an ant, to a fungus, that's okay, but it's not that useful. But what it induces the ants to do, it changes the ants' behavior, and it makes the ants grow up a plant or a tree and go to a fairly highest point it can, and then it goes in what's called the death grip. And as the ant dies, it grips, literally, it grips the central vein or the midribs of a tree, dies, and then the fruiting body explodes, releases the spores, and they go into the airstream and then decimate on the wind. So it's that way of dispersing the spores. And this film company have used this zombie fungus as something that they can possibly use to produce this series. Of course, people are now getting worried that this will happen in real life. Now, one last thing before I finish is murder, or as the Scottish used to say, murder. This is the death cap, Amanita phallides, and it's become, I mean, we know it kills people. We've known that for years. And if you're stupid, you don't pick it, but people do pick it inadvertently. People think it's a mushroom. It must be okay to eat. It's not. It's very deadly. We think just a quarter of that alone is enough to kill a family. So Australia, you've probably seen this in the press. It's still ongoing. She's denying everything, but they're determined to have her. And several doctors have testified in a trial of this Miss Patterson. And they've told them that the mushrooms in this dish that she prepared called beef Wellington, she said they were sourced from Woolworths and from an unspecified Chinese or Asian grocer. Okay. But the mushrooms were actually Amanita phallides. And they were part of this dish that she served up to her in-laws, as well as Gail's sister, and she served it to her husband, Ian, as well. Now, all of them suffered Amanita phallides poisoning. This was obvious, yeah. But the husband survived, and they think he survived. I mean, he only just survived, but they think it was survived because he didn't eat much of the meal. He wasn't very happy with the way it was. She wouldn't let anybody into the kitchen to help her serve up the meal. She served herself the same meal, but obviously she was fine. But when they looked at the evidence from her phone and CCTV, and they brought in a mycology expert, she was found in the area. Amanita phallides was found in the area where it was growing at the time of this incident. So it's not looking good. So I think she will be prosecuted for murder. And that's the end. And I'll just leave you with a mycology joke. And there you go. You should get it. It shouldn't need any explanation.